by defining what we mean by biopesticides very briefly. There's lots of different definitions. The Bateman et al. Paper published in 2021 includes 3 categories of biopesticide. Microbial pesticides, which are the ones that I'm most familiar with, which are things like bacteria, fungus, viruses that grow within the crop pest. Then we have macrobials, which are the parasitoids and predators, which we heard about in previous sessions. And then a broad grouping of biochemical pesticides, which includes the botanicals, as well as other things such as insect growth regulators and so on. This session is mainly going to be focused on the microbial biopesticides and the botanicals with some other biorational products. We've got a great lineup of speakers, so without more ado, we'll hand over to the first speaker. I should just say that you've got 10 minutes for each of your presentations, and I'll give you a nod at 8 minutes to start winding up if we get to that point. I think all of the presentations are going to be done by ICT in South Africa, so you don't need to share your screens, but Rhett will clarify that if needs be. Okay, can we go on to our first speaker, which is Dr. Nialangelo Acuna from the Philippines, who'll be talking about field efficacy validation of 3 entomopathogenic fungi as biocontrol agents against corn fall armyworm. Okay, thank you.
Okay, so thank you for this introduction. So may I begin now? Sir Ken?
We're hearing you, yes.
Yes. So I will begin by stating the background of the study. So the study is posted, the title of the study. So it is funded by our country's Bureau of Agricultural Research. Okay, so it is conducted by the Department of Agriculture, Bureau of Plant Industry, Crop Pest Management Division. So this study deals with actually 3 points. Okay, so first is the field efficacy validation itself. Okay, so the testing of Aisaria, Metarhizium, and Boveria bassiana against corn fall armyworm. And the other one is to establish the sustainability of the technology. This involves regional participation. And the last point is to publicly advocate the technology to the farmers. Okay, so this study is conducted because the use of chemical insecticides were the trend in controlling the corn fall armyworm in the Philippine setting. Okay, so it's hard to admit, but it is what it is. Chemicals are the way to go. So we are looking to harnessing something that is safer on the user, as well as more sustainable, since chemical use spells out cost. So there are many researches about entomopathogenic fungi controlling corn fall armyworm, but our technology has already optimized certain aspects, such as the preparation, the production of the fungi itself. Okay, so for that thought, we are looking into something that can be assessed in the field, and so that we can show the efficacy of our entomopathogenic fungi to the farmers for them to adopt easily the technology. Okay, so we have 3 NCBI registered entomopathogenic fungi. So this is packaged in the technology so that we can carry on to the control of the corn fall armyworm. So next slide, please. So it is significant blatantly because corn fall armyworm already noted a damage ranging from 15.11% to 32.5%. Okay, so that's significantly huge, considering that it is just a pest. So it may complex with other pests or diseases. So it needs to be dealt with accordingly. Next slide, please. Next slide, please. Excuse me. Okay, thank you. So the objectives generally is to verify the field efficacy of the three entomopathogenic fungi, if it is valid or not to be used on the control. Okay, so specifically, we will validate the percent control so that we will identify which is the best option to go. Second is to enhance capacity of technical staff. This is to establish sustainability so that the technology can perpetuate and can be used accordingly. Okay, so number 3 is to publicly advocate for the sake of public awareness. Okay, next slide, please. So the mechanism of it is we have 3 entomopathogenic fungi. We have from each of them we have 4 variations of spore concentrations. So 1 times 10 to the 6 up to 1 times 10 to the 12 with increments of 2 exponents. Okay, so these are the components and underlying practices embedded on it. Next slide, please. So upon validation, we identified that no significant difference occurred among entomopathogenic fungi, Metarhizium, Beauveria, and Aisaria. But we identified that 1 times 10 to the 10th spore concentration per ml, the best result. Okay, so these are the regional sites, and these are the percent control of each. Okay, so next slide, please. So these are the images of infection on site. So from left, we have Metarhizium infected cornfall armyworm. In the middle, we have Beauveria, and on the rightmost, we have Aisaria. Okay. So we have also observed residual effect in terms of total yield. So actually, it's not the total yield that has been affected, but the plant growth itself. So we have observed increments in growth performance with the use of EPF. Okay, so literature says also that the EPF has the capability to do so in terms of endophytic growth, solubilization of phosphates, and somehow also as extension of roots to acquire more nutrients. Okay, so next slide, please. So since the technology has been promising, we trained the technical staff from the regional crop protection centers so that they know how to manufacture and produce the biopesticide, as well as to distribute it to the clients, which is the farmers. Okay, so public advocacy was also conducted. Next slide, please. So predominantly, the concentration was 1 times 10 to the 10th. That's the most promising among the trials. So plant growth has been affected beneficially, translating to yield output increase, and training of trainers was conducted, and it is deemed necessary to have a sustainable fall armyworm control. Okay, so with that, I would like to wrap up the discussion with a thank you.
Thank you very much for a very interesting talk. I should say, if you have any questions, please put those into the Q&A box. We have a couple of minutes for questions if people have them. I think just looking at the questions that have come up so far, I think you've addressed most of them. One question is, how specific are the entomopathogens you're using? So what effect does it have on other Lepidoptera or natural enemies?
So first, I will deal with the question on the mortality of natural enemies. So pardon, I'm not able to show the images, but we took care of that. So natural enemies are generally not affected by the entomopathogenic fungi. So we've seen Coticia existing on the cornfield, but it's not affected by the EPF, as well as coccinellid beetles that are beneficial. They are not also, the EPF was also not infected, the natural enemies like that. So generally, no natural enemies was affected by our biopesticide. So, okay. In terms of concentration, yes, sir. May I address this one? So the concentrations, we use 1 times 10 to the 6, 1 times 10 to the 8, 1 times 10 to the 10, and 1 times 10 to the 12. So among those, the 1 times 10 to the 10 proved to be the best option.
Okay, good. We have one very quick question, which is, how do you maintain quality control of the fungi?
So time to time on the laboratory, we conducted assays. So we conducted infection to the cornfall armyworm, since we have breeding protocols. So we infect cornfall armyworm, and if it's not infective, then we will have other choices like returning it to the soil to retain its virulence, or another option is to reinfect to weak larvae, so that they can infect and then they can regain their virulence.
Okay, thank you. I think it's time to move on to our next presentation now. You can load up Angela's talk. This is by Dr. Angela Makindi from the Nelson Mandela Institute in Arusha in Tanzania. And she'll be talking about botanical pesticides use among smallholder farmers for fall armyworm management in Tanzania and Malawi. Thank you, Angela.
Thank you very much, Wilson, and thank you, everyone. Hello. I am presenting about use of botanicals on fall armyworms management, and our work is funded by the McKnight Foundation and is in collaboration with a number of experts from the NRI, from Q, Nelson Mandela, where I belong, and Luana in Malawi. So in my next slide, I am going to speak about botanicals in the general view, which are raw materials that are around in our environment, and especially in the African environment, and which can be used in a number of forms, such as being used as powders, as pure components, but could be processed as well. And they have been known by research results, but also by indigenous knowledge that they are very effective for crop pest management, but also for livestock and also in the storage. And they do that by a number of mechanisms. My next slide is speaking about their advantages. So being natural, some botanicals could also be used as medicines. So their harmfulness can be thought of in two different ways, and therefore not considered as lethal completely, because it has some broad spectrum uses and benefits, but also they persist less in the environment and therefore they have less toxic impacts, but they enhance ecosystem services, such as plants themselves could harbor natural enemies and pollinators, and therefore add some other agricultural benefits, such as natural pest regulation. But of more important in my talk also is the fact that these are substances that could be prepared locally. And this is very important because we are dealing with smallholder farmers who are really hustling with pesticides that are costly on one hand, but also they are sometimes could be adulterated, could be decanted into some containers that could also cause problems health-wise and to the environment. So I'll go straight to the work that we've been doing on fall armyworm, and this is the last year's experiment where we tried to examine or to experiment the locally processed botanicals and how they could be effective on fall armyworm management. So in this graph, the higher the effectiveness, these are the lower scores. And chili, pyrethrum are among the botanicals that were very good at controlling insecticides, and I mean controlling fall armyworm. And this is so promising to us. You can see here that we have powders and extracts. I'll say it in the forthcoming slides on why powder, why extracts. In fact, we had started with just applying extracts on a plant, on a maize plant, on a whole, so to say. And after some time, we had a number of farmers involved, and some opinions came that even a powder could be as useful, and we're seeing also powders being as effective. The next slide is from experiments from this year, actually, where we tried some demo plots as well as the farmer field plots. The far right is a farmer who is trying to apply some substances on a whole, but of interest is that in a demo plot, in this second picture, is showing a field having maize very well grown. There was so minimal fall armyworm infestation. So there was some infestation in the very early stage of the growth, and a number of botanicals were applied. Pyrethrum, neem were one of them, which worked quite very well. But with time, the choice of the demo plot made us understand that landscape diversity could have played. In fact, this must be the case. Could have played a very good role in minimizing the fall armyworm infestation. So this is the field that was not so much infested. They had minimum infestation in the beginning, which were controlled, but even in the negative control, there was so minimum infestation to the end. So we are learning that landscape would have a good contribution in this, and therefore a mix of using botanicals plus managing the landscape would really make a significant management of fall armyworm. My next slide is an on-station trial that was done to check. No, this is not an on-station trial. This is a farmer field trial where farmers used a different substance in addition to botanicals. This is a diatomaceous F. I thought it was wise to put forth this as one of the substances that has been used and has been successful in controlling fall armyworm. We are seeing less number of live fall armyworm, but also dead fall armyworm, and also less plants having frass. So showing that this was a good control mechanism. My next slide is on the on-station trial then, which was trying to check also a number of other botanicals. And here we are seeing tobacco, cymbopogon, and neem as botanicals that were having more efficacy as compared to the negative control. Of course, not as much as the synthetic pesticide, but much better than when it is not controlled, which is giving us a very promising and prospect of using botanicals, refining them better, finding good ways of processing them, finding good ways of applying them to obtain better results. So these are just results of the fall armyworm infestation in the early whole, so up to 10 days of maize growth. And the next slide is showing quite the same thing, but when the maize is a bit advanced in growth, so just before tasseling. We are seeing also again neem and cymbopogon and tobacco having better results in fall armyworm management. And we went ahead and tried to check on the impacts also of the botanicals on the yield of the maize. And the same botanicals that had contribution in the management of the fall armyworm had also some impacts on the yield, checking on the cob weight, on the length, on the grain weight, and things like that. This is in the next slide. Hello? Okay, thank you. Yeah, so this shows the contribution of botanicals in the maize yield, showing either less infestation, which would have caused the higher yield and something like that. So in summary, in the next slide, what we are trying to communicate here is like neem, pyrethrum, and cymbopogon are some superior botanicals that will be used for fall armyworm management. Although there is a room for a number of more botanicals that will be screened at local level to try and see their potentials. That is one. And although in the experiments that are presented, we have seen extracts and powder. Actually, farmers in Tanzania would prefer to use powder application to extracts. Of course, this is connected to the whole hassle of extracting, soaking, filtering, and all that. But it gives us also an insight that powdered materials would be also as effective as the other synthetic pesticides. And also, what we learned again this year, along with using botanicals, managing landscapes would also be an augmenting factor in order to make sure we reduce much the infestation. And also another observation is, using botanicals would also contribute into maize yield when checked into that perspective. Having said that, I thank you very much for your attention.
Thank you very much, Angela, for a really interesting talk and keeping perfectly the time. Can I remind you, if you have any questions for our speakers, if you can put it into the Q&A section rather than the chat, and then all the questions are in one place, and the speakers can answer those in the chat as well. Okay, we'll obviously at the end of this session we'll have a longer period for questions of about half an hour. So at that point we'll go through each of the questions and get some discussion going. Okay, move on to our next presentation, which is by Dr. Sachi Gurumayan. Sorry if I've mispronounced that, Sachi. He's going to be talking about enabling smallholder farmers to manage fall armyworm through an integrated approach in Africa and South Asia. Okay, so over to you, Sachi.
Thank you, Ken. Good afternoon, everyone. It's a pleasure to be here. Hopefully the slight transition will keep up with the pace. So I'm going to talk to you a little bit about Fauligen, which is one of our baculovirus products, one that we've been evaluating and trialing and also commercializing in many countries in Africa and South Asia, and some future plans around what we want to do in ASEAN. If we could go to the next slide, please. So Egg Biotech is originally an Australian company where, a few Australian scientists and farmers got together to combine nature, leverage what's out there in nature, apply our R&D skills, and then make it viable for a profitable and scalable agriculture. Because very often when you think about biologicals, you're not thinking about mass application. So we wanted to make that difference. In 2015, Payne Schwartz Partners took a strategic interest in the company through a significant investment and relocated the HQ to Texas to target the Americas markets, which is probably why you would have seen in recent years, months, our significant progress in the Brazilian market, for example. We have a number of products for key agricultural pests, such as the soybean looper, fall armyworm, corn earworm, Tuta absoluta, diamondback moth, and a few more in the pipeline that we will eventually announce and release. If we could go to the next slide, please. So our history has been one of continuous innovation, where if you see ever since the company was founded, we've been launching new products quite regularly, all based on baculoviruses, but with one or two that help in the application of baculoviruses. So magnet is a moth attractant, significantly reduces the population of moths, so that as you come in with a baculovirus product, it's more effective. Optimal helps to spread out. Now, if you look at the number of launches and geographic footprints that we've established since 2015, it is quite significant, and that's primarily coming from the strategic investment that the private equity group made. If we could go to the next slide, please. So if you're wondering what NPVs are, baculoviruses or nucleopolyhedroviruses, which is essentially what we base our products on, they are naturally occurring viruses that are specific to a particular species, for example, the fall armyworm species. Now, what we do is we harvest those viruses and formulate them into products. They normally occur, the DNA of the virus occurs in protein crystals called occlusion bodies, which once ingested, break down in the stomach of the caterpillars and release the virus DNA, which then sort of recognizes the host caterpillar, enters the body cells, and then multiplies. And that process, it weakens the caterpillar and then eventually liquefies and kills it. But in that, that essentially enables the beneficial insects that are out there to target, to go after the caterpillars directly. The other thing that is interesting about fall armyworm is that because they're cannibalistic, the strong and the healthy ones will go after the weak and the infected ones. And in doing so, they end up getting infected themselves. So there is this multiplier effect out in the field, which helps us to control the pests for a longer period of time. Of course, fall armyworm is extremely mobile, so there will always be new population coming in. Now, the unique mode of action that NPVs have enables us to make it a part of any IPM program with significant results. We have not known or had any cases of resistance against baculoviruses in all the years, in all the decades that we've been using the product on a commercial scale. And we're talking millions of hectares in Brazil, in Australia, in the US, and so on. This also allows flexibility for the farmer in terms of when to spray, because the product's non-toxic, non-hazardous. It degrades quite fast out in the open environment, in direct sunlight and high temperature. So it offers quite a lot of advantages, enabling the farmer to almost have a very small pre-harvest interval, if any. And that helps us in managing pests as well as residue and so on. If we could go to the next slide, please. Now, this is Fauligen. It's a soluble concentrate, as you see there, and it's based on the fall armyworm NPV virus. Now, because of the fact that, fall armyworm, and as I described before, is very specific to the fall armyworm caterpillar, it doesn't affect the the natural ecosystem that's already there. It's certified organic in the US. It has been proven. I'll show you some results as a standalone or as part of an integrated pest management program. And as mentioned before, no non-resistance to NPV products. If we could go to the next slide, please. So the history, I mean, we had been managing fall armyworm in North America and Brazil for a while when fall armyworm spread to Africa. So we stepped up our efforts to support African farmers. We were already in Brazil. So when the African delegation came, led by USAID, we were there. We established partnerships with CABI in Kenya, IITA in Nigeria, CIRAD in Senegal to cover the sort of different agroecological environments in sub-Saharan Africa to evaluate foliagent, because we wanted to know that this product would work in the African environment as well before we trialed and commercialized it. And we also started trials in multiple countries. Early 2019, we announced a partnership with the FAO and USAID in South Sudan, targeting thousands of farmers. I will tell a bit more about that later on. But same year we started, we continued with the trials in a number of countries in sub-Saharan Africa. Interestingly, we were also introduced to Isfahani in Bangladesh by USAID and CIMID. So that was one of the fastest evaluations, trials, and registrations for foliagent because of the sheer pressure of foliagent, of fall armyworm in the country. We also partnered with Bowers in Sri Lanka, a Swiss legacy company that helped us to accelerate its adoption among smallholder farmers in Sri Lanka. We now have a partnership with UPL for sub-Saharan Africa, where we've been working with them to distribute, make product available to multiple smallholder farmers in the right pack, right size at an affordable price, for example. So now it's registered in multiple countries. Earlier this year, you would have probably noticed that we announced a partnership with Golden Agri, which is one of the group companies of flour mills of Nigeria. And it was also registered in Kenya. And we continue to work with stakeholders to emphasize on its benefits and how it can help smallholder farmers manage fall armyworm in a safe manner. We're scaling it up, of course, and as fall armyworm has spread to Southeast Asia, I am now in multiple discussions with stakeholders and potential partners in the Philippines, Vietnam, Indonesia, and the region in general, to talk about how we evaluate and commercialize this in the region. If you could move to the next slide, please. Next slide, please. Hello? Hello?
We have the next slide, please.
The slides are not, oh, there he goes. Yeah.
Thank you. Oh, so just to give you some data, in all the countries where we've been evaluating and continue to evaluate foliagent against untreated controls or chemicals that are popularly used to control fall armyworm, what you can see there is that foliagent performs as a standalone in this particular case on its own against some of the well-known chemicals. It continues to deliver good yield and can work as a product on its own. You also see the picture there, it speaks 1,000 words to see the difference between the control and foliagent treated product cobs. Now, the thing is, it's a biological, so you have to scout regularly, you have to apply it at the right time. And because these are noctics, we prefer that you spray it late in the evening or at sundown, so there is time for it to be ingested. And more so at an early stage of the caterpillar. So, stay instar 1, 2, 3, ideally, because once they grow bigger and stronger, it's more difficult for the biologicals to have an impact. So scouting and spraying at the right time, right stage of the caterpillar are critical to make it effective. If we could go to the next slide, please. So I mentioned it also works as part of an IPM program where you can mix it. But in this case, what we've seen in Brazil is that when you combine it with chemicals that are beginning to have issues of resistance or have their own performance, but you want to extend it, we're seeing significantly better yield, as you can see there in both corn and cotton, by combining Cartogen, which is the Brazil brand name for foliagent, with the chemical. This is the protector concept, which we believe is enhancing the performance of chemicals. So one one way to combine it there. The next slide, please. This one's talking about what I mentioned earlier on about the partnership in South Sudan, where we had a PPP with USAID, the Ministry of Agriculture, FAO, being on the ground, and worked with CIMID and CABI, which provided training of the trainers, the extension agents at its facility in Nairobi, Kenya. And we were able to establish demo farms on the ground to train farmers at different sites through different clusters in each of the communities that we were targeting to help with the training and adoption. Interestingly, because of COVID, we couldn't really organize the demo farms that we planned, this year. So we ended up using an adapted video from the Scientific Animation Without Borders. They have a whole bunch of videos on fall armyworm, scouting, and so on. So we adapted one of those, put them on tablets or smartphones, and used that to train in small groups. In other parts of the world, in other emerging markets, let's say, we are developing business models through strategic partnerships with distributors on an exclusive basis. For example, we work with research partners, some of which you can see there, international collaborators. I mean, the idea is to address the issues around awareness. Do farmers know about such a product? If not, how do we train them? How do we make sure that they're aware of it? Availability, of course, that's one critical thing, at the right price, at the right pack size, for example, designed for their benefit, and affordability. And we are working on all of this with our distributors. And I'm quite pleased to say, I mean, it's gaining traction, certainly in Africa and South Asia, and we intend to repeat this in ASEAN countries as well. I think that's if you go to the next slide, it's just a thank you.
And I was just about to ask you to wind up, so maybe we'll just finish.
Yeah, I'm just waiting for the next slide, Ken. I'm done.
Yeah.
It's not moving.
No.
Could you please go to the next slide, please?
Okay.
I'm done anyway.
Yeah. Well, thanks for a really great talk. I think it shows the potential that public-private partnerships could have in terms of enabling the adoption of biopesticides. There's lots of questions coming in, which we'll address at the end. Lots of people interested in this and want to know about its availability and the constraints of using these biopesticides. But for now, we need to move on to our next speaker, which is Dr. Krepan Anewanyu. Apologies again for my linguistic skills. We'll be talking about the bioefficacy of the Artemis Earth household soaps and neem oil against Spodoptera furder larvae in Benin. Okay, so over to you, Krepan.
Okay, thank you. My name is Krepan Anewanyu. I am a PhD student at the Faculty of Agronomic Sciences of the University of Abu-Mekarabi. So I'm very pleased to share this finding with you, which is entitled Bioefficacy of the Atomic-Source Earth and Other Biorational Products in the Management of Fall Armyworm in Benin. Next slide. Okay. Since the invasion of the pests, several damage occur on maize crop, and the main reaction of farmers was based on the use of chemical insecticides. And sometimes the overuse of these products can generate another problem such as resistance and a risk on human health and also on the environment. So there is now a need to develop an alternative control method that are based on the use of biopesticide and also natural enemies. Next slide. So in our research, we investigated firstly the larvicidal activity from some locally available soaps and detergents. And the ones which is produced in our country are named Pamida soap and Koto. And after that, we have the detergent Clean, which is commonly available in our country. So we use the immersion method using the second instar larvae of fall armyworm, and the different concentration of these soaps we evaluated range from 0.5 to 2% concentration, and we replicate these tests 10 times under laboratory condition. Next slides. Okay, the result indicated that under laboratory condition, all the soap solutions evaluated were effective in killing the second star larvae of fall armyworm, but Palmida soap, the one which is locally made in our country, promoted the highest mortality rates, and we estimated its lethal concentration, which is also lower compared to others. So from this result, we have moved to field where we investigated the efficacy of this soap with other biorational products. Next slides. Next slides. So experiments were conducted in two different agroecological zones of our country. The first is located in the northern part, and the second one in the south. And we use a randomized complete block design, and with 4 replicates and 5 treatments. So the extra precocious variety of maize was used during this experiment, which was conducted from 2019 to 2020. Please, next slide. So among the different products we have tested are the Palmida soap, the one which promoted the higher mortality rate under laboratory condition, which is tested at 0.5% concentration due to phytotoxicity that can occur when the concentration is above. And after that, we have the neem oil, which is also tested, and the diatomaceous earth provided by Imerys, a firm which is located at the United States of America, which is tested at 2 different rates, 7.5 and 15 kgs per hectare. And then we also tested the chemical insecticide, which is commonly used in our country by farmers, imametine benzoate, at the rate of 0.6 liter per hectare. So these different products, except for the chemical insecticide, were applied weekly from 21 days after sowing until to 42 days after sowing. Please move to the next slides. Okay, so the result indicated a significant reduction in the population of fall armyworm and also the percentage of damaged plant. Because I forgot to mention earlier that during the experiment, we have recorded the number of fall armyworm larvae per plant before each spray, and then the damage severity, and also the population of natural enemy. So in the left side, when we look at the plot of the number of fall armyworm larvae per plant, we observe that the higher population of fall armyworm from 28 days until 42 after sowing were recorded in the untreated plot in both experiment sites. And at the same time, we also recorded the lowest population of the fall armyworm larvae in plot treated with the Palmida soap and the chemical insecticide. And concerning the percentage of damaged plant, we also observe a higher percentage of damaged plant in the untreated control, while the lowest were promoted in the first seat place by the local soap, and on the second side, it is the chemical insecticide which promoted the lowest damage, the lowest percentage of damage plant. So move to the next slides. Okay, we also perform on cost benefit analysis to see whether the application of this product is profitable for farmers or not. And we will observe that at the first site, experimental site Ajun, the chemical insecticides promote the higher yield and also generate a profit for farmers with a net gain which comes out up to 166%. But when we move to the second experimental set, Ndali, we observed the higher yield, which is promoted by the diatomaceous earth, and which generates a more profit for farmers with a net gain up to 54% compared to the untreated plot. I'm sorry, in this table, I've made a mistake because I report the yield of diatomaceous in the place of in the place of the chemical insecticide. So I'm very sorry for this mistake. So please to the next slide. Next, please. Okay, in summary, we realized that the rational product tested in this study were effective in the management of fall armyworm, and some of them produce similar or better results than the chemical insecticide, which is commonly used by farmers. But from this result, we have to move to the next step, which is to implement a collaborative research with farmers in the sort of learning group in order to tailor this technology for scaling up, and then to investigate the potential of over agroecological management approach, such as the management of soil fertility and the weeds on the control of these notarial insect pests. So I think I am at the end of my presentation, and thank you for your kind attention.
Great, thank you very much, Kleppen. That's a really clear and informative presentation. Again, questions are coming in, but we've reached our time, so we'll move on to our next speaker, and we can catch up with those questions at the end of this session. Hopefully our next speaker is here, Dr. Hailu. He'll be talking about the elucidation of the science and effectiveness of local innovations for managing fall armyworm in Uganda. Over to you.
Thank you so much. As you rightly mentioned, I'm going to talk the local effectiveness of local innovations for managing fall armyworm. This study was conducted in collaboration with NARO, National Agriculture Research Organization, and ECP. Next slide, please. So I don't want to dwell a lot on the biology or distribution of fall armyworm. We heard a lot about it, and there's a lot of effort being done by different scientists with regard to the management of fall armyworm, like agroecological management options. Several options are coming. Biopesticides are being validated in different locations, which are also effective. Host plant resistance, we heard a lot about this effort in the previous sessions, but to ensure the IPM is well executed, it's also important to consider local innovations or indigenous technical knowledge, which can be evaluated and incorporated into IPM recommendations for fall armyworm management. So the objective of our study was to determine effectiveness of selected local innovations for the control of fall armyworm under lab and field conditions. Before we move into these activities, there was a farmer group discussion conducted in different districts in Uganda, and farmers gave us different options like kerosene, livestock urine, plant extracts of different species. So we selected few of those ones to evaluate. Let's move to the next slide, please. So the laboratory observation was done in Kawanda Research Station, Naro, whereby most of these lab observations were conducted with the relative temperature of 25 to 28 degrees Celsius and relative humidity of 65 to 67 degrees Celsius. So these experiments, the laboratory experiments were conducted like two tests, which I'm going to present here. One is a choice test, the other one is a no choice test, and seeing the development of the larvae. All the observations were conducted on CRD design. Let's go to the next question. So for the choice test, what was done was leaf cuts of about 5 grams were selected and kept in a petri dish, whereby about 10 semi-star larvae were released at the center to be given an opportunity to move to whichever plant material they prefer to move. I didn't mention the treatments for these observations, so we identified neem, lantana, camara, detergent, and ash. So all these were assessed to see the larval movement at 1 hour after the release, and then 48 hours after the release, and monitored which direction the larvae were moving. Next slide, please. Next slide.
I think it's frozen again.
Oh, maybe I think let me put off the video. The internet is too slow.
I think it's actually on their side, on the
yeah. So the second experiment was the no choice test, whereby we released the larvae into either neem treated, lantana treated, or water treated and ash treated leaf cuts of maize, and then we observed the larval development and then mortality.
Sorry, we're changing computers. Just hold on a little bit. Okay, thank you. You may proceed.
Okay.
On the right slide, right?
Yeah. Okay.
Okay.
So to determine the effectiveness of the local innovations, as I said, the larvae were released at the center of the petri dish and monitored on the larval development. This is for the no choice test. So we can move to the next. So similar observation was done under field condition. These experiments were conducted in 2 districts in Uganda, and the treatments were neem leaf extract, lantana leaf extract, detergent. In this case, we didn't use the powder, we used the soap, soil, water, and insecticide control. The experiments, the treatments were replicated 3 times, and observations were done at 3, 6, 9 weeks and 12 weeks after planting. Let's move to the next. So the findings, we've observed that the fall armyworm larval orientation showed more inclination to the control, to water treated, while the ash, detergent, lantana, and neem treated leaves were not chosen the same way as at the control. The lowest was the neem, but interestingly, the detergent was found to be more attractive for the larvae, and we've observed more larvae moved towards the detergent treated leaves. Otherwise, ash, lantana, and the neem showed less number of larvae moving to towards the treated leaves. Then 48 hours later, can you move it a little bit up? Yes, please. Thank you. So 48 hours later, still the detergent was more attractive to the fall armyworm larvae, while less larvae moved to lantana and neem. When it comes to ash, they were relatively the same. So they started. The effect reduced after 48 hours. Let's go to the next one. So in terms of now, under the no choice test, larval mortality, interestingly, although they were very much attracted to detergent applied the leaves of maize, most of the larvae died on a detergent applied leaf extracts. Ash was 80%, while neem, the larval mortality was 10%. As you can see that the larvae were able to stay alive on the neem treated and then lantana treated leaves, while all of the larvae died on the detergent. When it comes to pupation, it reduced significantly. Neem treated leaves, larvae fed on neem treated leaves did not pupate that much, 20%, while lantana treated ones were 50%. Detergent, since the larvae died, there's no pupae, and we can move to the lower one. Yeah, thank you. So adult eclosion, adult emergence, when it comes to the detergent and ash, all died. The water treated 70% adult emerged, the neem only 10% emerged. So even though the larvae were feeding on neem treated leaves, but very few were able to emerge. So it has a considerable benefit when it comes to reducing the successive generations of fall armyworm. We can go to the next one. Under field condition, the effects were not that much visible, the same way as we had in the lab observation. Insecticide treated in Bulambuli district was lost at 29%, while the control was 46%. As you can see, the level of infestation did not differ much with the rest of the treatments in Tororo. No, I mean in Bulambuli district, while the same was observed, but relatively the same was observed in Tororo districts as well. So the infestation was slightly higher, but here the major observation that we had was the time of treatment. It's hot, so the treatment application probably was supposed to be done in the evening, so we are not able to see much difference, although the laboratory observation showed us some variation with it. But farmers were very much keen about this. We did it on a participatory approach. Farmers were evaluating also the process, and some of the suggestion also came from the farmers that the time of applications and proper scouting is also very critical. We did it at 369, but it's better to conduct a good scouting so that the effect of the extracts were visible. The next slide, please. Okay, so from what we observed, the neem leaf extract showed great potential to manage fall armyworm, especially if the formulation and the time of application were redesigned. It would have been much more effective. The fall armyworm fed on neem treated leaves were unable to pupate. As you have seen, only 10% pupated in the lab observation. That means it has the potential to reduce the successive generation. The use of local innovations to be effective, there is a need to determine effective formulation and the frequency of application, as I said. The participatory approach of field experiment encouraged farmers to use local innovations as an integral part of IPM. We observed that further studies need to evaluate effectiveness of detergent since it showed provide significant mortality in the lab. But during the in the course of the day, there was also a discussion regarding its toxicity or effect on soils and all this. So a comprehensive study is needed. We strongly believe that the local innovations can be strengthened to be part of fall armyworm IPM. I think that's all from my side, but if I have saved some time, my colleague Winnie might also come in and say a few words.
I think we're running a bit over time now, so we'll hopefully our last speaker is there. I can't see them. Do we know if Joseph is online? Maybe Rhett or Lydia could tell me what the status is there. Rhett?
Sorry, I was just checking on who it was. I have not seen them. So if they are not, let me see if we can find them amongst the so if the last speaker could identify themselves to us, it would help.
Dr. Joseph Mniwoki from Isiphi, Kenya. Anybody else from Isiphi know whether the speaker is in attendance?
I think, Ken, maybe you just proceed with some questions and then we'll, if we find them, we'll come back to you.
Okay, thank you. Well, I'd like to thank all our speakers to begin with, and some really interesting stuff going on in the sort of biopesticide, biorational field. Before looking at the individual questions, I'd like to maybe pick up on some general issues, and the main ones that always come up with biopesticides are the cost, relative cost compared to conventional chemical pesticides, the availability.
Talk?
Sorry?
Oh, sorry. No, this is the last talk. It's my thing. Sorry.
The availability in country. We've heard about some locally available detergents, for example, things like ash as well. But what about the more traditional commercially available biopesticides? And Shachi will no doubt have something to say on this from AgBiotech. Though I should mention that there are other commercial companies producing biopesticides out there, and Shachi from one company that came forward to give a presentation. Oh, we have a presentation. Do we have a speaker? Continues. So availability and cost are two of the main things. There's also the issue of repeatability, replicatability. Sometimes these things work, sometimes they don't. So what I'd like to do is ask the speakers for, from their perspectives in the different experiments that they've done, to maybe comment on those three things in terms of the cost of the various things they're looking at, the availability of them, and the efficacy of them and how repeatable that is. So who'd like to speak first?
Ken, I'm just thinking maybe it's better to remove the presentation if the speaker's not there, so we are actually...
Yeah, I agree. There we go.
I don't know if anybody wants to go first on the...
You might as well go ahead now. You've started.
I mean, I think, you know, in terms of whether the results are replicable, I think I'll start with that, because one of the reasons why we had that partnership with CABI, IITA, CIRAD, and others is because we wanted to see if we would get similar results in sub-Saharan Africa. And then, you know, when we got similar results here, good performance against fall armyworm, you know, and comparable, if not better in some cases, against what is out there as standard control, we also went and did the same thing with the Bangladesh Agricultural Research Institute, with the Wheat Maize Research Institute in Bangladesh, and then again in Sri Lanka with the Department of Agriculture. And we've seen that, you know, it works. And this is where I think, you know, from a biological industry perspective, where AgBiotech stands, we need to be able to produce products that are consistent in quality and performance. And that means that, you know, we have to have that center of excellence for as a production facility where we rear caterpillars. I mean, during peak season, we rear somewhere between 1.2 to 1.5 million caterpillars a day at the factory. So it just tells you the scale at which we operate. In terms of availability, I mean, with our distribution partners, we are working in every sense possible. So for example, in Bangladesh, we have small sachets. So the distributor imports in bulk and repackages it into small sachets to make it more affordable, more accessible for smallholder farmers. In parts of Africa, it's available in 100 ml bottles because that is the standard size that they would use for the farmland that they have. It varies by country, so we try to accommodate that. And then affordability, I mean, we make it, you know, we make it at such a large scale, you know, volume wise, to make it affordable, because we're taking advantage of economies of scale with Fauligen and all our NPV products, which means that by the time it's translated into a farmer price, after the distributors have added their margins and the retailers, everybody has to make some margin along the way, it remains quite affordable. And we've seen that, you know, compared to, for example, in Kenya, compared to the standard we tested our product against, our ROI is actually quite good. It can be over 100%. So I think I can't give you the individual prices by country because that is determined by the distributor in the country. But what I can say is that it's very affordable. Thank you.
Okay, does anybody else want to comment from their perspective? Maybe Angela or one of the others who've been working at the small scale farmer level.
While we wait, just to give you an idea, that's the sachet from Bangladesh.
Krepen, you want to say something?
Okay, so I just want to make some precision about the locally available soaps we have investigated. So further research are going on presently with farmers in order to evaluate the way we can use these soap without damaging the population of natural enemies, and also without no phytotoxicity on these leaves. So I think that's my last words.
Okay. Just general point on that. What do you think is the mechanism of action of the detergent?
It acts just like by contact. They disrupt the permeability of the spiracle of the larvae, which finally dies from hydration. I don't know if I'm clear. It acts just as a contact insecticide.
Okay.
Yes.
Germa, would you like to comment?
Well, with the bar soap, I doubt it if there is an insecticidal property, because it's an emulsifier. Probably it prohibits the movement on the feeding behavior. We use the detergent under lab condition, the powder, but there is also a question of toxicity and the effect on the plant itself and the soil. So we opted to use the bar soap just to prohibit the feeding behavior. We did not expect it to have insecticidal property from the bar soap.
Presumably these are readily available products, that the soaps and detergents. One of the advantages is readily available to farmers, correct?
Yes, they are available. Both are available. I think the most important thing is in quantifying the amount when it comes to applying the detergent. Since it is also applied in the maize hull, it might not even have that much effect on the soil, because a little amount is sprayed targeted to maize hull. The most important is the important thing is to be careful about the toxicity or the damage that may cause to the maize itself, not to the soil as such.
Yeah. Niall, do you have anything to add? Philippines?
Okay, I think, can I go ahead?
Yeah, I was hoping that Niall
from the result we got now, we are moving to the investigation of agroecological approaches, such as the management of soil fertility, and also the way we can manage weeds in maize crops in order to reduce the infestation of fall armyworm. That's what we are targeting now in our future researches.
Okay. Another issue related to this is farmers' perceptions about the effectiveness of biological pesticides and various other things. And I think this research to show that a lot of the issues are associated with having had bad experiences with biopesticides previously, or not knowing what a biopesticide is, or what the difference is between a biological pesticide and a chemical pesticide. Based on your experiences, perhaps you could comment on that, maybe starting with Shachi.
Thank you, Ken. I mean, it is a serious concern, and it is challenging because people are so familiar with chemical pesticides and their mode of action. They associate biologicals to do something similar. Yet we all know that biologicals behave and act in a different manner. So we need to work on the education. So for example, the work that we did in South Sudan, there was a lot of hand-holding along with our partners, because we wanted farmers to know that this is a different product. You have to use it differently. You have to wait to see actions, because some of our farmers were so used to spraying chemicals and seeing dead insects by the time they come back, after lunch. And we said, no, it doesn't work like that. You spray it at sundown, you wait, and then you let it ingest. And then in a few days, you'll see that the caterpillars are not working. They're not quite as active anymore. And then you'll see dead caterpillars a few days later. And that is, it's a question of risk as well. Farmers want to see that the caterpillars are taken care of immediately and they feel secure. So we have to work on that to make sure that they are aware of the product, that it will work, and build that confidence one season after another. Thank you.
Anybody else want to comment on that?
I think it's a very important issue. It is also unwinding what we have done to farmers, because it is us, the researchers, who recommended effective insecticides, knockdown effect, and everything, efficacy. So farmers are now used to that kind of results, not knowing what kind of environmental impact it will have and all this. And then the advertisements and agrovet companies and all these are also playing a significant role for farmers to choose the chemicals that are showing them results immediately. I think participatory approach, showing the farmers what kind of effect it does and why we are doing this in this way, and then acknowledging the fact that the training that we gave them before was not considering much, and then the amount of pesticides that we are applying and then the risk it is associated to it is enormous. So farmers can get it. Farmers will recognize the role of the natural enemies when they see them on their own eyes. Otherwise, they recognize all insects as bad insects, except few ones. So our experience in training farmers under field condition showed us that farmers are they also understand, because they were spraying chemicals for long, and if the effect is literally zero. So moving to biological control, our farmer friends, showing them the insects that are helping them in the field, the biological controls, why biological controls are taking time to kill the insects, and killing them slowly but surely is the best way approach. So we need to redesign our training manuals, trainings approaches to address the farmers. We need to retrain the extension agents who are reaching farmers more frequently. I think if we do that, we'll be able to change that mindset of using synthetic insecticides.
Very good point. Yeah. Krepan, did you want to add anything?
Okay, I think no, it is good.
No.
Yes.
Okay. So one of the other issues with many biopesticides, including the baculoviruses, is that you need to target the early life stages, as Chachi alluded to. The same is true, I think, of the fungal products and maybe the bacteria as well. Maybe those of you who've been working on botanicals and other biorational products could say something about those issues with those products. Are they most effective against the younger stages, or can they be effective against the larger stages as well? Gurma, do you want to kick us off?
Okay, we did the biopesticide validation trials for fall armyworm management. Our observation is they are much more vulnerable when they are at early stage. Secondly, immediately after hatching, or they are moving down to the maize hole, it's easier to apply and then reach them, so that it can be effective before they have developed, before they are grown. So from our small observation under field condition, the earlier stages are much more vulnerable than the mature ones. And again, farmers will also not buy it if the damage already happened and you kill the larvae, because now they see that the maize is already affected. So if it's done early enough with a good scouting, it's a lot easier to manage, and they are also more vulnerable at early stage.
Anybody else want to add to that? So in the previous session was all about synthetic pesticides, and what I took from that session, which was also really interesting, was actually how complex it is to determine how to apply even synthetic chemicals in terms of getting the dose right, getting the timing right, when to apply in terms of time of day and so on. And I guess those issues are magnified to some extent even by biological pesticides, because particularly with the microbial biopesticides, you've got that extra added complexity, which is the biological properties of the biopesticide itself. Does anybody want to sort of comment on that observation?
maybe I can make a, I mean, one of the things that I mentioned earlier is, it's taking advantage actually of the pest's behavior. So if they're noctuids, you go and spray at a time when they would be out feeding. And that's what we recommend for our farmers, at sundown when it's beginning to cool down. One of the questions one of the many questions I should say is about storage stability, and I'll address that a bit as well. But in terms of timing of the spray, you don't want to spray it at at midday when the sun is right above you, because that will deactivate the virus quite rapidly. However, if you apply it at sundown when the temperature is cooling down, there's less sun and the pest is slowly beginning to come out, you take advantage of its natural feeding habits to make it ingest the virus. And then once it gets going, it activates this chain reaction where one infected caterpillar will spread it to others, and there is this natural multiplication. I think so. It's about educating and working with partners and stakeholders to work with the ecosystem rather than just controlling everything or eliminating everything that is out there that could be damaging to the crop. Do you mind if I touch a bit on the storage stability, because that's a question?
Okay. Yeah.
So one of the things about biologicals is that we do need to take care while handling them. And this is true for most biologicals. And I would say for, in fact, 90% of the products that are out there. So for NPVs, for our baculovirus-based products, what we do say is, minimize exposure to temperatures of 40 degrees or to direct sunlight, because that will have an impact on the product. It will not deactivate 100% immediately, but it will lose its activity. The other thing is, if you want to store it, you can store it in the same manner you would normally store seeds to maintain quality, because seeds are living organisms at the end of the day. And so, if you store it in an air-conditioned room or in a shaded area at the back of the retail shop where the temperature maybe is in the range of 20 to 30 degrees and there's no direct exposure to sunlight, this product will last for seasons. If you want to keep it forever, we've had experiences where we had products in the deep freezer for 18 years. We brought it out and then tested it, and it was just as efficacious as 20 years ago. So I think storage is a critical part, but I think with education, not every biological product has to be stored in a refrigerator, even at the last mile outlet. That's what I'm trying to say.
Okay. Does one of the other speakers want to talk about botanicals and the other biorationals in terms of shelf life and storage and efficacy? No.
I guess not.
But presumably botanicals, having used neem previously, the efficacy of that will decline fairly rapidly over time once it's been prepared, certainly in aqueous solution.
To be honest with you, I mean, as someone who trained as a formulator, though many years ago, I mean, even chemical formulations are not stable when you expose them to direct sunlight. We might add stabilizers and so on to keep it, stable for a while, but it's not recommended practice anyway.
Just a comment here from George Ferry saying, later this afternoon, he'll be sharing results from their experiences with botanical aqueous extracts. So stay tuned for more on the botanicals, I think. Just scrolling through the questions. Most of these questions will be and have been answered by the speakers in the Q&A session. So I don't want to repeat that too much. I guess one other theme that is coming out in terms of the number of applications and how that compares for the biologicals and botanicals compared to the chemicals. Goma, maybe you could kick us off on that.
Okay, thank you so much. I think application or management appropriate scouting. Specifically for fall armyworm, we've done some activities like with the community-based fall armyworm monitoring. So if we do the monitoring, comes starting from egg crushing up to application of different botanicals or insecticides or biopesticides. So when it comes to the effectiveness, we've seen we did spray that 3 weeks after planting, 9 weeks after planting, but this from what we observed is not the right thing. It's very important. First of all, the fall armyworm life cycle population is too many. So if you apply now, in 3 days, they will start laying their eggs. So when you come back, the situation is different. So if we train farmers, or if we do proper scouting, and then we start application of the biopesticides, immediately when we assume that majority are hatching, then it will be effective. So maybe even one application immediately after scouting. But farmers also need to know the situation, whether there was rain, whether it was a completely dry spell. From what I have seen in the field, rainfall is a good control measure because they are feeding on the maize hole, and the maize can keep water in the maize hole, so they can easily drown and die without even applying a pesticide. So we need to factor in the environmental factors if there is a need to apply. And then applying in the evening is a lot better. For experiment purpose, we go out during the daytime and we apply. So the subsequent heat can also disintegrate and reduce the effectiveness. So application time and proper scouting are the major things to be done to be effective. So the reputation depends on now based on the scouting that we are doing. In some cases, 2-3 generations affect. In some cases, there's even more. Sometimes it's early infestation, which is very, very critical, where farmers also neglect. When maize has only 2 leaves, the fall armyworm can finish them. Infestation, when it is very visible during vegetative growth stage, maize can easily compensate the loss, and then still farmers may get yield. Infestation at silking stage, one larvae caterpillar feeding on the silk can feed on the silk, and the whole crop can be. So there are critical times when this application should be done. So I think we need to look into all these factors to be effective with the biopesticides or botanicals that we are applying.
Open, did you want to add something?
Yes, I just want to mention that the efficacy of biorational should be take into consideration the level of infestation before the spray. So we have to check this level based on the action threshold first, and then when we reach it, we can spray. But the great challenges we are meeting about the biorational product is the amount of solution to be sprayed, because they are not synthetic. So they require more volume of solution to be effective. So that is the great challenge we are meeting with farmers. But we recommend, based on the level of infestation, a weekly application of this biorational product in the case the infestation level is very high. So that's what I can say.
Thank you. Oh, we're just about to run out of time. So I just
do you mind if I just add a comment on the number of sprays?
Yeah.
Because one of the things that we have seen with Phologen, having tried it in multiple parts of the world, is I think it depends a lot on the ecosystem as well. So in some countries where the fields they may be smallholder fields, but they're interconnected, and so it is a vast open space where the pest can just fly and spread, or even swing from one plant to another, or crawl. you do need a higher number of sprays. But what we've also seen in places like Sri Lanka, for example, where smallholder plots are, are divided by or separated by banana plants and palm trees and coconut trees and so on, we've seen that you can get away with 1 or 2, maybe 3 maximum sprays, whereas in other parts where you need 5, 6 sprays. The other thing is, if you time your first spray well, such that the product, the NPV, is ingested by the caterpillars, remember, each caterpillar then becomes a biofactory producing billions of particles. So even though the product itself may not, be compatible with sitting in direct sunlight for long, each caterpillar that has become a biofactory is quite stable. So those are things that we need to work with. Thank you.
Okay, thank you. And on that note, I think we'll draw it to an end. Peter Juwada makes a number of good comments there about this issue of armyworm moving between plants, and the fact that because that is happening, then you will need more spray applications. But also, I think a more general comment is that people working with biological pesticides need to really think more and stronger about application methods and approaches, and not just to think about botanicals and biologicals as being the same as chemicals in terms of how you apply them. And I think combining approaches in an integrated pest management framework is the way forward for the long-term management of fall armyworm, including safer synthetic chemical, but also biologicals and biopesticides, so that we can allow the natural enemies to build up and to help do the control for us as much as possible. And on that hopefully positive note, I will draw this session to an end and just ask whether Rhett has any comments, any issues he wants to raise before we finish.
No, not at all. Thank you very much. It was a very interesting session, and I think I learned a lot. So thank you very much, Ken, for chairing the session, and thank you to the speakers for giving us such an interesting set of talks.
Yeah,
so
really interesting.
We will. So the next session starts again in 9 minutes. So yeah, you can take a little break, and then we'll be back with the last session of the conference in another 9 minutes.
Thank you.
Thank you, everyone.
Thank you.